Welcome to class of high throughput data generation and analysis. Students, today we are going to study about the transformation of expression ratio. So what is expression ratio? We have discussed about this in the last lecture. So expression ratio is the spot intensity ratio in the two different conditions. So spot intensity of test sample is divided by the spot intensity of reference sample. So that is the expression ratio RK by GK. So it is denoted by TK. TK is equal to RK by GK. Expression ratio is a relevant way of representing expression differences in a very intuitive manner. Okay, what it means? Okay, for example, if genes do not differ in their expression level, so that will have the TK. What will be the value of the TK? That will be 1. TK is equal to RK by GK. So expression ratio will be 1. Means there is no difference in the expression level of two genes. So when we compare the expression of the test sample, the gene expressed in the test sample and with the reference sample. So why we need the transformation? Because for the more understanding we need the transformation. So the expression ratio of 1 shows no change in the expression, and the genes show the similar expression, but this is unhelpful. This may not align with the data if the genes are upregulated and downregulated. Okay, so this is not the useful information. This is not the useful prediction. Okay, for example, a gene that is upregulated, we can upregulated by a factor of 4. Means expression ratio of RK by GK. So 4G divided by G. So that is 4. The ratio is 4. So the expression of the gene is upregulated by the factor of 4. That means genes are highly expressed. This value shows high expression of genes. So on the microarray slides, the spots that will have the more intensity, so they will show this expression, means the spot that will show the high expression. So those genes are highly upregulated. So there may be some other case also where the gene is downregulated by the factor of 4. What it means? Now the expression ratio become 0.25. Means what is the formula? TK is equal to RK by GK. Okay, R by G. That means R by 4R. So the ratio is now 1 by 4. That means 0.25. So it shows the down regulation of the genes. Means genes are showing low expression. The expression is low. So thus the up regulation is blown up and mapped between 1 and infinity, whereas the down regulation is compressed and mapped between 0 and 1. So this is called mapping. So is this useful? So we can think this is useful or not. Up regulation is mapped to 1 to infinity, and down regulation, that is mapped, means 0 to 1. So genes that are showing the up regulation, that are mapped between 1 to infinity, and genes that show the down regulation, which show the down regulation, means low expression. So these are mapped between 0 to 1. So in this you can see the inconsistency, means the upregulated genes that are showing 1 to, that are mapped from 1 to infinity, but in the down regulation we are mapping it to 0 to 1. So one can perform the two. So to remove this inconsistency, now one can perform the two different types of transformation of expression ratio. What are these transformation? Inverse transformation and logarithmic transformation. What is the problem? Because 1 to infinity, that is mapping, this is very difficult to analyze those genes. And on the same graph, if you want to draw the graph on the page, okay, so it will be different. There will be no difference. If you want to see the difference between the genes, the expression level of the genes, so it is very difficult. I will show you the figure. So this may be like this. In this figure you can see, like in the first one figure, okay, because mapping space is very large on the first one figure. So here you can see, we are unable to see. Without the transformation, we are unable to analyze the data. So this is the problem. Okay, so to eliminate this problem, we apply the two different methods of transformation: inverse transformation and logarithmic transformation. So what is inverse or reciprocal transformation? In this method, the mapping space is discontinuous. Although this is also applicable, but mapping space is discontinuous between -1 to +1. Okay, so what happened in this method? Genes that are mapped from 1 to infinity. So now here in this method these are mapped up to 1 plus 1, means 0 to 1. Genes that are showing the high regulation, that are up regulated, so these are mapped in the mapping space from 0 to 1. Okay, that means genes are showing the up regulation. Now the genes which are showing the down regulation, that are mapped between 0 to -1. So this is called inverse or reciprocal transformation. So initially we were not taking it in account, minus value we were not taking. Here you can see, so for the down regulated gene we were using 0 to 1, value between 0 to 1. So this was the problem. But in the reciprocal transformation, we are using the value below the 0, means that is -1. So initially we have not considered that, means we have not taken the mapping space like that. Now this is useful also, because the genes that shows the up regulation, that are mapped from 0 to 1. Okay, means 1 to infinity. Now we have limited the mapping space from 0 to 1, and for the down regulated gene means 0 to -1. So in this, now the mapping space is very small. So here we can see, we can map. Okay, now it is easy. Easily we can analyze the genes. Now the fold change, you can see TK is the expression ratio. If TK is greater than 1, greater than or equal to 1, so it will be mapped mapped. Okay, it will be shown by the value of 1 only, and the fold change. Okay, now if there is a ratio is the -1 by TK, if TK is less than 1, so that will be mapped below the 0. Okay, but it will not go beyond the -1. So in the up regulation, it will not go beyond the value of 1. So value will not be greater than 1. And for the down regulation, value will not below the -1. So fold chain we can deduct 4 and -4. So when the TK is equal to 4, and in the case of the down regulation, the TK will become 0.25. Okay, for the -4. So here you can see, this is called the reciprocal transformation. But there may be some problem in the reciprocal transformation also, because mapping space is discontinuous between -1 to +1, hence become a problem in mathematical. But this is also the useful method. So what is another method? Logarithmic transformation, a better transformation procedure. So comparatively, logarithmic transformation is thought to be considered best method. So what happens in this is to take the logarithm of base 2 value of the expression ratio. Okay, like log 2 expression ratio. So what is the benefit of this? Because in the calculation, you use the logarithmic table. Why you used to take the logarithm? Because if the values are large, so the calculation will become easy. It becomes easy to calculate the large values. So to minimize the mapping space, we used to do that. So these are the major advantages, that it treats differential up regulation and down regulation equally. Also has a continuous mapping space. Okay, how it happens? For example, if the expression ratio is 1, then log 2 equals to 0. Means if there is no change in the expression, if the two genes are showing the similar expression, means the genes of the test sample is showing the expression with the reference sample. That means, so the value of the expression ratio will be 0. Means no change in the expression. If the expression ratio is 4, then log 2 equals to plus 2. Okay, whatever the value, means for the upregulated genes, it will be means it will not go beyond the 2. For the expression ratio of 2, 1/4, so it is equal to -2. So now you can see the mapping space is from -2 to 4. For the down regulated gene, it will go up to -2. For the up regulated gene, it will go up to +2. And 0 is 4. There is no change in the expression of the genes. In this transformation, the mapping space is continuous and up regulation, down regulation are comparable. So here in the figure you can see this is the logarithmic transformation method. This is the useful method. Okay, so logarithmic transformation makes the distribution of microarray data symmetrical and normal and fits. Okay, so Gaussian distribution method is used here. This is the example of that. In the first figure, you can see x-axis and y-axis. So the values are given on the x-axis. So these are the large value. So here you can see on the axis, I mean the values are clustered towards the axis. Okay, origin. So when there is no transformation, but after transformation, what happens? Now here you can see the logarithm of base 2 that is applied. Data is transformed now. Now the data has been transformed. So here you can see, I will just explore this. So in the first one figure, you are not able to see the difference. You are not able to distinguish what is the differential expression. So here in the first one figure, you can see these are the large values. The mapping space is large. Now we have minimized that. So here you can see the values are minimized now. On the x-axis you can see, and you can also see the peaks and the expression of the genes. It varies. So clear cut difference. You can distinguish at least. So there are the two, the genes are shown in the two colors, like group 1 and group 2. So there is a maroon color and blue color or purple. Okay. So but when we are taking the logarithm of base 2, so this is the figure, this is the transformation. So here in the first one figure you are not able to analyze anything. In the second figure you are seeing, you are able to analyze. Okay. And after the transformation of when we are taking the logarithm of base 10, this is the figure. Here you can see. Okay. Again, and this is the different logarithmic transformation when you are taking the logarithm of base e. Okay. So these are the two. There are the two groups: group 1 genes, group 2 genes. So here you can see the differences among the result. So after applying, after the transformation, now you are able to analyze. You are able to see the differential expression of the genes. Okay. So the mapping space. So data has been transformed and there is a mapping space has been used that we can analyze. So why we are minimizing that? Means so that we can analyze easily, because 1 to infinite is a very difficult. One will be only the person will be involved in the calculation. It is very difficult to calculate to analyze the expression of the genes when there is a large number of genes. There is a large data set in the biological process, in the bioinformatics data, so that is very large. From the microarray slides we used to get the expression data of the thousands of genes. So microarray is the method in which we can analyze the expression of the genes simultaneously, the thousand of the genes, the differential expression of gene. What is the differential expression? Means genes are expressed, same gene is expressed in the different condition. So we can analyze the expression of the same gene in the different conditions. Like we can take the two groups, means the group of means test sample or reference sample, and we can compare that. We can compare the expression ratio. So these were the all two methods that can be applied. So transformation of expression ratio is useful because here we are trying to minimize the mapping space, because 1 to infinite and 0 to 1, this is the inconsistency. For that we have applied the two methods of transformation, that is called inverse transformation and logarithmic transformation. So today we have discussed about these two methods. These are helpful. So mapping spaces means -1 to +1 in the reciprocal transformation. So this is also the easy method, but the mapping space is discontinuous. But in comparison to reciprocal transformation, logarithmic transformation is considered more useful, because here we are applying the logarithm and the mapping space becomes like -2 to +2. So for the up regulation value is +2, and for down regulation -2. Okay, and 0 means 0 value shows there is no expression. Sorry, there is no change in the expression, because here we are comparing the two groups of the genes. And what is the meaning of the expression ratio? So we are analyzing the expression ratio. So it's become easy after the transformation. It becomes easy to analyze the expression ratio of different genes, thousands of genes or large data set of genes. We can compare the expression ratio. Okay, so these are the useful method. These are the notes. So from these are the source where I have taken, and I have taken the one figure, this one from the another slides. Okay, so now lecture is over.